Hi, my name is Quentin Tradup, and I'm a fourth year at Augsburg College. I'll be pursuing a degree in biology with Chem minor. And today I'd like to talk about how the immune system interacts with the central nervous system during a bacterial infection. So, to start off, I'm going to give you guys a preview of what I'll be talking about today. To start, I'll be talking about the central nervous system, giving you a brief overview. And then I'll lay into the immune system and kind of how the factors are like the key players here, and then talk about the blood-brain barrier, and then how the immune system interacts with the central nervous system, and then to finish, I'll talk about a bacterial infection inside the brain and what's occurring. All right, to start. The central nervous system consists of a brain and spinal cord, and the brain was considered to be immune privileged. And what this meant was that during a graft, the brain had a very low rejection rate, and so essentially the immune system wasn't able to pick up that the graft was foreign, and essentially this meant that the immune system was unable to interact with the central nervous system. This was later disproved, and I'll talk about that later. Now, the immune system consists of multiple organs that produce multiple cells, and these cells interact within the body to prevent infections and keep you alive. Now, some key cells I'll be talking about today are microglia, oligodendrocytes, astrocytes, neutrophils, T cells, dendrites, monocytes. Now, I'll give kind of an overview. I'll talk about their functions later. Now, in the immune system, there are two sub-branches, the adaptive immunity and the innate immunity. And now, when we get an infection, the first thing that happens is the innate immune response. And the innate immune response is fast-acting, but is not specific and has no memory. So it's really inefficiently effective, I guess is the way to put it, where it is able to act quickly, but with no direction or aim, so this isn't good in the brain. This is not what we want to happen in the brain, but we want this pretty much is just to make sure, hold off until the cavalry arrives, essentially, or backup. And the backup would be the adaptive immune response. We want the adaptive immune response in the brain. What the adaptive immune response is it consists of having memory. It's specific, diverse. It's a little bit slower than the innate immunity, but it's effective, and it has lasting effects. And another key thing, it's able to determine self versus non-self. And those are all very important players when we're talking about the brain, infections in the brain. So how do we get an innate immune response to shift over to an adaptive response? Well, I'll talk about that later. Now, before I jump into the blood-brain barrier, we're going to talk about something that's occurring in the immune system, and that is neutrophil rolling. Neutrophil rolling is where neutrophils are attracted to a site to the site of infection via chemokines, and the neutrophils, once at the site, begin rolling. And when they're rolling, they're interacting with the surface of the molecule, the surface, and by interacting with the surface, they're touching integrins and they're touching selectins and integrins on the they're interacting with the surface and touching selectins. Now, when they're touching selectins, they slow down and begin to adhere to the surface. And once they adhere to the surface, they're able to become activated via surface proteins on the neutrophils, integrins. And once they become activated, they're able to transmigrate through the endothelial cell junctions into the site of infection. Now, once they're in the site of infection, the neutrophils can do their job, and I'll talk about their job later. Now, the blood-brain barrier consists of a network of capillaries made up of capillaries. Now, these capillaries consist of endothelial cells with tight junctions. On top of those capillaries, they also have astrocyte feet, pericytes, and microglia. The blood-brain barrier allows passive diffusion of waters, some gases, and lipid-soluble molecules. On top of that, the blood-brain barrier also acts as a primary defense against bacterial infections for the brain. Now, the brain and the blood-brain barrier were thought to be immune privileged sites, and due to the selective permeability, of the blood-brain barrier towards the immune system. Now, there was research done that initially confirmed this, but then there was later research done that deconfirmed this. So let's talk about the first research. The first research done was staining of the circulatory system and putting dye into the circulatory system. And once the dye had ran its course through the circulatory system, they examined the organs, and they found that most organs were stained, except for the brain. And this led them to believe that the circulatory system was being prohibited or not able to interact fully with the central nervous system because of the blood-brain barrier. And so essentially the immune system wouldn't be able to interact with the brain because of the blood-brain barrier. Now, they did a graft test on the brain and found that with one graft, little to no rejection rate occurred. So this further confirmed that the immune system was unable to interact with the central nervous system. But then somebody had the idea to do 2 grafts. And by doing 2 grafts, they found that the second graft was rejected immediately. And this led them to believe that if it's rejected, that means the immune system was able to detect this foreign body as foreign, and so therefore the immune system is able to interact with the central nervous system. So now the blood-brain barrier isn't this impenetrable defense we thought it was. So now we had to relook at all the things connecting these 3. So the immune system interacting with the central nervous system. In the case of a neural inflammation, the blood-brain barrier begins to change its conformation, and chemokine production increases, as well as an upregulation of surface adhesion molecules. And essentially what this means is that T cells and other immune cells are able to infiltrate the central nervous system. So while all these cells are coming in, cellular recruitment is still being managed. So essentially they're limiting how many cells come in so that an autoimmune response doesn't occur. And this is thanks to the glial cells, which promote apoptosis of our cells. So now that I've kind of talked about an interaction between the immune system and the central nervous system, let's talk about interacting when there's a bacterial infection. Now, a bacterial infection of the brain is rare, but they're almost always fatal. So there's something to be reckoned with. We need to really kind of focus on them. Now, these bacterial, how these bacterial infections get inside, they occur by 3 mechanisms. The first one being direct destruction of the capillary endothelial cells. The second one is disruption of the tight junction gaps, and therefore allowing bacteria to get in between the endothelial cells and into the site. And then the third one is the bacteria are able to utilize vesicles and get across the membrane and into the area. Now, once the bacteria is inside, the innate immune response kicks off right away. So now that the innate immune response is occurring, we need the adaptive immunity to come and save the day. So in order for that to happen, some cells need to come into play. The first one is microglia, which phagocytose and eat bacteria, and when they eat bacteria, they break it down and present it to other cells as antigen, and this allows for the cells to kind of remember what to look for and have a sense of direction. And so essentially the first part of adaptive immunity is occurring here. And besides that, they're able to kind of break down the blood-brain barrier and allow more cells to come in, more immune cells to come in. Now, astrocytes are the partners to microglia. They essentially do everything the microglia do, but under normal conditions, they actually prevent one of the ways bacteria are able to infiltrate to the brain. They essentially contribute to gap junction stability. Sorry. And so the bacteria are unable to use method 2. Now, in the case of infection, they, like I said earlier, assist microglia in everything they do, but also release chemokines that recruit neutrophils, monocytes, and T cells, and also upregulate surface adhesion, allowing more cells to come in again. So essentially, key thing here is the amount of cells that are coming in, making it a fast and effective way to kill bacteria so that neuronal damage does not occur. So now that all these cells are coming in, among those are neutrophils, and neutrophils are your primary host defense against infection. And what they do is they phagocytose everything, and they are really good at it. Besides that, they also upregulate adhesion molecules, bringing in more of their buddies to come and kill the bacteria. A curious thing that's occurring here is that the blood-brain barrier expresses selectins that the neutrophil is able to interact with and do neutrophil rolling. So it's essentially the blood-brain barrier wants neutrophils to come in towards the infection. So it's facilitating the immune response. Neutrophils have a curious thing where they release defensins, lytic enzymes, and also antimicrobial peptide. And what this is it's your defense once the immune system has killed the bacteria. It's to make sure that the bacteria doesn't come back again for round 2, essentially. Another thing neutrophils do is they release chemokines that shift the immune response from an innate to an adaptive response. And when we talk about adaptive, we're going to talk about T cells. And what T cells do is they produce interferon gamma, which kind of promotes the generation of chemokines that attract monocytes and T cells, which essentially means that the adaptive immune response is in full flight here. It's occurring fully. And essentially now the bacteria is losing the fight. So to cover, let's go over what I've covered today. During an infection, the immune response, the immune system signals for more cells to come to the site of infection. To start, an innate response occurs, but then is shifted over to an adaptive response. The blood-brain barrier becomes more permeable as it interacts with immune cells and essentially facilitates the immune response. All this is occurring quickly and effectively so that neuronal damage is minimized. And the immune system has checks and balances to make sure that an the autoimmune response doesn't occur. So the glial cells, in this case, apoptosis. And in conclusion, we can see that by observing how the immune system interacts with the central nervous system via the blood-brain barrier's facilitation for the immune system, we can determine that the blood-brain barrier and brain aren't as immune privileged as we thought they'd be, and now the blood-brain barrier is actually more permeable when it interacts with the immune system, and this allows the immune system to interact with the central nervous system. Some future research would be to better understand the interaction between the central nervous system and the immune system, so that we can maybe kind of target certain infections in the brain, such as MS, and hopefully come up with a cure. At this time, I'd like to open the floor to questions.